The relationship between the gut microbiome and insulin resistance is rapidly emerging as one of the most exciting frontiers in nutrition and metabolic research. Far from being a simple digestive organ, the gut acts as a metabolic command center, and the trillions of microbes residing there are active participants in glucose regulation. Clinical studies increasingly show that the composition of the gut microbiome, the metabolites it produces, and the way it interacts with dietary inputs can meaningfully influence insulin sensitivity. This means that diet is not just a source of calories, it is a tool for shaping microbial communities and metabolic outcomes. Certain microbial compounds can exacerbate insulin resistance, while others can actively enhance glucose metabolism, and the difference often comes down to the type of foods consumed. Emerging dietary trials highlight the central role of short-chain fatty acids, SCFAs, bile acid metabolites, and polyphenol-derived compounds in mediating these effects. SCFAs such as butyrate, acetate, and propionate are produced when fiber and resistant starches are fermented by gut bacteria. Clinical evidence indicates that these molecules help strengthen the gut barrier, reduce systemic inflammation, and improve insulin signaling in the liver, adipose tissue, and muscle. For example, trials with high-fiber diets or prebiotic supplementation have demonstrated measurable reductions in fasting insulin levels, improved HOMA-IR scores, and enhanced postprandial glucose control. These benefits appear to be mediated largely by changes in microbial composition, favoring SCFA-producing genera like Roseburia, Lactospiraceae, and Bifidobacterium. Participants in such trials not only experienced metabolic improvements, but also reported better satiety and reduced cravings, illustrating the holistic benefits of aligning diet with microbiome health. Conversely, diets high in refined carbohydrates, added sugars, and ultra-processed foods tend to feed gut microbes that produce pro-inflammatory metabolites. Lipopolysaccharides, LPS, for example, are bacterial components that can leak into the bloodstream when gut barrier function is compromised, triggering chronic low-grade inflammation, a known contributor to insulin resistance. Clinical studies show that reducing processed carbohydrate intake while increasing fiber and polyphenol can decrease circulating LPS levels, improve markers of systemic inflammation such as CRP and IL-6, and enhance insulin sensitivity. This demonstrates that gut-derived compounds act as critical mediators between diet and metabolic outcomes, and that the microbiome is highly responsive to dietary interventions. Several recent trials have also highlighted the importance of polyphenol in modulating insulin resistance via the microbiome. Foods such as berries, nuts, green tea, and cocoa provide polyphenol that are transformed by gut bacteria into smaller, bioactive metabolites. These compounds appear to improve insulin signaling, reduce oxidative stress, and enhance SCFA production. For example, a study supplementing participants with polyphenol-rich food showed shifts in microbial populations toward beneficial taxa, higher SCFA output, and measurable improvements in fasting glucose and insulin levels over several weeks. These findings underscore that dietary inputs can shape microbial metabolism in ways that directly influence host glucose regulation. The cumulative evidence from these trials suggests that insulin resistance is not merely a product of calorie imbalance or genetics. It is a dynamic process influenced by the gut microbiome's metabolic activity. By providing the right substrates, fermentable fibers, resistant starches, polyphenol, and unsaturated fats, diets can promote the growth of microbes that enhance insulin sensitivity while reducing the abundance of microbes that produce inflammatory or insulin-disrupting metabolites. Even small, consistent dietary adjustments can shift microbial composition, SCFA production, and inflammatory signaling, yielding measurable improvements in glucose regulation over weeks to months. Beyond metabolic markers, these trials indicate broader, systemic benefits. Enhanced SCFA production not only improves insulin sensitivity, but also strengthens gut barrier integrity, reduces endotoxin exposure, modulates appetite, and may influence energy expenditure. Polyphenol metabolites reduce oxidative stress and systemic inflammation, while balanced microbial communities support more predictable circadian rhythms and metabolic signaling. Collectively, these mechanisms illustrate that diet and microbiome are in continuous dialogue, and by optimizing this interaction, individuals can actively shape their metabolic trajectory. From a practical standpoint, these findings provide clear guidance. Diets that emphasize whole, Minimally processed foods rich in fiber, prebiotics, nuts, legumes, vegetables, fruits, and polyphenol are likely to foster microbial communities that promote insulin sensitivity. Conversely, frequent consumption of refined sugars, processed snacks, and high glycemic foods can drive the proliferation of microbes that contribute to inflammation and impaired glucose metabolism. Aligning dietary choices with microbiome health is thus a tangible, evidence-based strategy for improving insulin sensitivity, reducing the risk of type 2 diabetes, and supporting long-term metabolic resilience. In essence, recent dietary trials highlight the gut microbiome as a central mediator between food and metabolic health. Gut-derived compounds, whether SCFAs that improve insulin signaling or pro-inflammatory molecules that impair it, are shaped by what we eat, when we eat, and in what combinations. Thoughtful, microbiome-friendly dietary patterns provide a powerful lever to enhance insulin sensitivity, reduce inflammation, and stabilize blood sugar, transforming the gut from a passive participant into an active partner in metabolic health. Over time, these dietary interventions can create a self-reinforcing cycle of microbial and metabolic resilience, offering both immediate benefits in glucose regulation and long-term protection against chronic metabolic disease.